Good evening, sir. Myself, 𝐀𝐝𝐶, Assistant Professor in Physics from Great Institute of Technology. It gives me immense pleasure to introduce Dr. Narayana Reddy, sir, Assistant General Manager, Kimberlite Chemical Private Limited in India. He completed his master in polymer science from Sri Krishnadevaraya University, Anantapur. Sir has served as researcher for 9 years in polymers, synthesis of emulsions, adhesives, design, and characteristics of materials. His keen passion involved in developing new products, enhancement, and applications for companies from different sectors. Sir has a potentiality of organization skills managing multiple assets to reach goals in a timely and cost-effective manner. He has remarkable accomplishment on various syntheses like acrylic dispensants, thickeners, and acrylic emulsions, biomedical applications, and so on. Sir has played a vital role in various responsibility like development of water treatment chemicals, ISO documentation, alternative development, vendor, and alternative development worked as assistant manager R&D at 𝐀𝐝𝐀𝐿 Industries Private Limited, Mumbai. R&D officer, automotive coating divisions, Mumbai, and research associated at University of MONS, Belgium. Sir has presented 22 national papers and international as well as also reviewed for journals. He has worked as an investigator and co-investigator for first HRD project with outstanding titles. Sir has also handled several key positions as R&D officer with Nippon Point Indian Private Limited, Mumbai. As postdoctoral fellow in Italian Institute of Technology, Naples, Italy. We are indeed very pleasant to have you, sir, as our resource person. Welcome you, sir, and we request you take over the session, sir.
Okay. Thank you, Dr. Shiva, for giving some brief introduction about me. So I'm very happy for you to giving. I'm very happy to give an opportunity to get an opportunity to provide some of research incentives, research activities during my PhD and postdoctoral attempt. So yeah. So
Thank you,
today
sir.
I'm going to actually have two sessions. So both sessions I'm going to give present on the different types of research activities I did during my PhD activities. So yeah, first session I'll start with polyacrylamide hydrogels for different applications. So yeah, so the first session. So are you getting my screen?
No, sir. Please
Yeah,
share your
wait.
screen.
Yes. Let me share my screen. Is it okay now?
Is it now okay, sir?
Yeah. Okay. So, good evening, all. So, basically, so I did my MSc in polymer science and then PhD also in polymer science from Sri Krishnadevaraya University from Anantapur, Andhra Pradesh. So, yeah, let me start with my first sessions with the evaluation of biocompatibility and drug-related studies of gelatin-based magnetic hydrogels. Okay. So yeah, so the present topic involves the development of hydrogels and then combination of nanoparticles which are developed inside of these hydrogels. Then we call in a hybrid network which is called hydrogel magnetic nanoparticles. So then these applications we are going for a different type of applications. So maybe we will go for the for drug delivery studies and then we go for the biomedical studies. So yes, so before going to this topic, so let understand is what is exactly hydrogels. So hydrogels is a one type of a three-dimensional. It is a one type of polymers. So where you can prepare by developing by converting monomers into the polymers by different type of polymer techniques. So then the polymer forms, but the polymers compared to regular polymers compared to hydrogels, it is one type of a cross-linked network. So where these polymers will have some cross-linking structure, so there it will when we dissolving into the water. So it will hold a lot of amount of the waters. That's why this name referred to as a hydrogels. So which when this polymer in kept in the water, it will absorb into the water and it will form a jelly nature. That's why it is called hydrogels. So based on this, it will absorb a lot of amount of water ranging from 10% to in thousands, number of times, thousands of their dry weight, so that it will use for multiple applications. So moreover, so why these hydrogels are used? Say in every sector, whenever developing some materials, ultimately it should meet some requirements. It should useful for some application purpose. So these hydrogels are also used for different type of applications. You can count into the baby diapers, you can count into the medical applications, you can count into for the water treatment, you can take into the any type, wherever you are going for regular applications, everywhere it will count. So these hydrogels will use for number of applications. So how these hydrogels are developed? As I said earlier, so if you take into the monomers, so these monomers, when you are treated with some catalyst system, then these monomers convert into the polymers, then you will get very giant network. So this giant network will act as a polymeric hydrogels. So with this, you can develop it by either copolymer system or homopolymer system or polymer system. It's depend based on your requirement, based on the types of choices you can select. You can develop different types of hydrogels. So some people use it to develop with natural polymers, some people use it to develop with synthetic polymers, some people use it for the combination of both natural as well as it's as I said here, based on your requirement, based on your end application. So the monomer comes into the picture. As per that, the final application will also decide. So in this way, you can develop these hydrogels. So these hydrogels, when you fix it to the certain environment, when you fix it to the certain external environment, so it will accordingly. For example, if you developed hydrogels and if you after you keep into the open atmosphere, or if you keep into the air environment, even if you go into dry environment, so during that time, this hydrogel again started to string. So whatever the material inside of the hydrogel, that will comes out. Maybe either it is a water or it is a something different solution that is absorbing. Whatever the material it is absorbing, that will comes out of the environment. So that will use for the different type of applications. So it is like a externalized stimuli responsive polymers also. So means when you are applying some external environment into the developed hydrogels, it will start to string and give some of the whatever the material it is holding. So it will be useful for different applications. So this is the basic concept of hydrogels. Okay. So yeah, I said like where it exactly it goes, this hydrogels, where it exactly goes. So these hydrogels will go for the agriculture and horticultural applications. So this again, it is going for the baby diapers. Even it is used for construction materials. It is used in the water blocking tapes. Then it is used for the fruit storage buildings and vegetables. Even artificial snow also it will create. Even that some of the electronic applications like gel actuators and sensors also it will use. Then medical waste treatments, and this again more importantly in the biomedical applications. So lot of areas it will be. So if I go for one by one, where it exactly agriculture applications? Agriculture application mainly it will use for the where water scarcity is more. In that area, you can use this agricultures very extensively. So for example, when there is a less rain, so when you are planting one tree, and if you keep your agriculture hydrogel near the plant, and if you pour the water, so it will absorb the water. So after certain time, when there is no water facilities available, so this hydrogel will release that whatever the amount of water that is hold, it will release into the environment. Means here the environments where the plant is available, plant is there. So this plant take the water from your hydrogel and it starts to growing. So in this way, hydrogels are useful for the agriculture applications, majorly for horticulture applications also. So again, then baby diapers also. So where your baby diapers also, because these hydrogels have a tendency to absorb the water. So whatever the human waste coming in from the babies, so if you keep in the diapers, it will absorb the water and it will hold it. So that will be you can throw it. So again, in the construction materials. So construction materials, so when you are going for the very big like metro construction, home, very big apartments, see, lot of water used to come into the that area. So to prevent this, so what these people, construction people, use it to attach this hydrogel to that environment. So it will arrest the water, so that there is no obstruction into the for construction work. Again, the same phenomena it will apply for even water blocking tapes also. So this again, so they are used for also the fruit storage buildings also. Yeah, and then sensors and actuators also. Some of the now recent developments, some of the hydrogels used for the some sensor purpose also. So then these hydrogels are also used for the biomedical applications. So biomedical application is the biggest area where hydrogels are used in verily. For example, your drug delivery applications, or even some catheter applications. Wherever some drug releasing requirement is there, so there you can use your hydrogels. So yeah. So what is again, see, since our work is developing involves the synthesis of some magnetic nanos composites and hydrogel network. So we are going to give some brief introduction about the magnetic nano composites also. So what is exactly magnetic nano composites? See, uh, we are already, we everybody, majority people knows that there is some nanoparticles available in our sector. So these nanoparticles are actually, it is they have different type of applications, but this is actually how it forms. So if you take some metal elements like iron or silver, some other etc. So when they are in, when their ions are converted to the zero valence state, so you will get some nanoparticles. So that nanoparticles are very great interest. So since our work involves in the developing of hydrogel magnetic nanocomposites, so we are taking up magnetic nanoparticles for our applications. So these exactly magnetic nanoparticles have very big like hydrogels. These also have very big application in different industries. So they are using for catalyst purpose, biomedical purpose, magnetic resonance imaging, data storage, and even environmental remediation. Environmental remediation, it's also one type of water purification technique. So where you can remove all the environmental, I mean, some metals that is polluted through some lot of the industrial waste, that you can remove from the magnetic nanoparticles also. So these nanoparticles have different characteristics, different properties, but majority where you will get some good advantage. So if they have the particle size is below 10 to 20 nanometers, means that will be very big useful for us. So wherever these particles, so many varieties to become nanoparticles, means maybe some higher nano size, some lower particle. But if you are getting very low nanoparticle size, that the application will be something more useful for your applications. So this these nanoparticles again, so it should be some super paramagnetic behavior. So when super paramagnetic behavior, then you will get some specific targeted applications like magnetic therapy, MRI scan. There you will get more advantage. So and behave like a so these magnetic movement and behave like a giant paramagnetic atom with fast response to applied magnetic fields with negligible remains and coercitivity. So that means, so what is actually super paramagnetic behavior means? See, some magnetic particles always behave their magnetic nature. So I mean, without external environment, without applying any field, it will always show magnetic nature. But super paramagnetic behavior means when you have applied some magnetic field, then only these particles will respond. If they are not if after removing your external magnetic field, it will not respond any magnetic behavior. So this is also one type of characteristic properties that where we have developed this super paramagnetic behavior. So based on this characteristic nature, they are using for a different type of applications. So 1 minute. So sorry, where this exactly these magnetic nanoparticles are used. So magnetic nanoparticles using for the magnetic recording media, catalysts, and ferrofluids, used as agents for magnetic resonance imaging and therapeutic agents for cancer treatment. So useful in optical switches and tunable diffraction gratings, employed in sealing computer disk. So uses in NMR probes for oil prospecting. So even some high surgery also, these nanoparticles are recommended to use. So based on this, so these nanoparticles have received considerable attention in the development of immunoassays. Immunoassays means like a protein suppression, protein purification, like that also it will be useful. So magnetic resonance imaging. So where your MRI analysis, everything will do through magnetic nanoparticles only, and even targeted drug delivery. So if something when people, patients like a cancer patients, so when they are going for the medical diagnosis, medical treatment, so if you inject a drug along with the magnetic nanoparticles, so you will get very targeted therapy so that you get only treatment where your real tumor exists. So so that rest of your body system will be saved from your side effects of your drug. So in this way, this magnetic nanoparticles are very helpful for us. So yeah, so since we are talking about the hydrogel magnetic nanocomposites, so what exactly it is? See, one side we are developing hydrogels, one side we are developing magnetic nanoparticles. So when we are bringing both these together, so you will form like a composite system. So where you have a dual system in your network. So one is hydrogel and another one is magnetic. So these magnetic nanoparticles we are developing into the inside of the hydrogels, so that you will get a composite complex system. So where it acts as a very strong composite system, strong polymer system. So that is called hydrogel magnetic nanocomposites. So how it forms? So these nanoparticles are incorporated to the hydrogel network. So thereby you are getting hydrogel magnetic nanocomposites. So that is some example, like you can say ferrogels. So this you can develop, you can prepare, you can develop in different type of routes. Like you can do co-precipitation technique, like or you can do in hydrothermal microemulsion or in biopolymer method. So many varieties is there where you can convert, where you can develop, where you can convert, where you can develop this hydrogel magnetic nanocomposites. So again, what is the application of this hydrogel magnetic nanocomposites? See, we have already selected applications for hydrogels, selected applications of nanoparticles. So when you are bringing these together, you can combine, you can get multiple applications of this hydrogel magnetic nanocomposite. So these are used for the hyperthermia applications, for control drug release, and for triggering thermosensitive changes in the hydrogel volume or shape. So in this way, exactly this person will go for the targeted therapy. So and this you can go for even for MRI analysis also. So when you are preparing this hydrogel magnetic nanocompose, when you are in this system, when you are into injecting into your body, so wherever the tumor is available, wherever tumor is there in your body, so it will exactly show the proper image of the your body, proper image of the tumor, so that you will know that whether it is a really cancerous tumor or benign tumor. So this will give your, I mean, exact clarity of your system. So this is one type of applications of hydrogel magnetic. So this is restricted for this. I mean, this is specifically using for biomedical applications. So again, apart from these mobile applications, so they are using for like nanometers, sensors, and variety of separation devices. Even like I said, for water treatment, water purifications also. So some of the industries, they are taking nano catalysts, where it should be maintained through the efficiency of the catalyst should be maintained throughout the reaction. So if you are properly incorporated nanoparticles in the hydrogel composite system, so the nano catalyst, the nanoparticles will play a good role to maintain the efficiency of the catalyst system. So you can have multiple applications, multiple opportunities for your hydrogel magnetic nanocomposites. So yeah, so far we have discussed about the hydrogels as well as hydrogel magnetic nanocomposites. So why we have taken up this work for this our studies? See, so that's why we are. I'm referring MNPs. MNPs, it's a short term of magnetic nanoparticles. So hereafter, if I said MNP means it's a magnetic nanoparticles. So where these magnetic nanoparticles, when it incorporate into the hydrogels, you will get a hydrogel magnetic nanocomposite. That's we are referring hydrogel magnetic composites. So these are used for the controlled release of drugs, mechanical devices, artificial muscles. So in defense as sensors, optical switches, and data storage systems. So okay, so far we are discussing about the applications and advantage of the hydrogels and hydrogel magnetic nanoparticles. So, but how will achieve these applications? See, wherever you are developing nanoparticles, irrespective of type of nanoparticle, whether it is a magnetic nanoparticles, whether it is a silver nanoparticles, whether it is a copper nanoparticles, whatever, wherever you are developing nanoparticles, so it should always maintain a good stability. It should always maintain a good stability. If it is stability in the sense, it should uniformly distribute. For example, I developed 100 nanoparticles, 100 number of nanoparticles. So if I developed 100 nanoparticles, since these nanoparticles have a tendency to bind each other and to form a big cluster, big like a stone form. Okay, so big stone form. If you if you bind like this, the phenomena, the application side, the results will be very, very bad. So you don't get your proper application. So means when you are developed 100 nanoparticles, it should uniformly distributed. Means one nanoparticle like this. It should be in separately, it should be distributed. Then only each nanoparticle acts as a individual magnet so that you will get very better performance. But these nanoparticles have a some I can say steric effect because they have some charges on their surfaces. So normally chemistry wise, like reject, opposite charges attract. So like this also, these nanoparticles where they will find the opposite charges and they will start to binding together. So they will form some big agglomeration. This is, I mean, this chemically or physically it is called agglomeration. So if you maintain the agglomeration, you don't get these properties. So to maintain the uniformity, to prevent the agglomeration, so lot of scientists, they are trying what they are doing. So they are coating these nanoparticles with some. The people are coating some of the chemical substance on the nanoparticle surfaces. So nanoparticle surface, so that what will happen? It will arrest the charge stabilization, so that each particles will separate, so that it will exist as a individual magnet. So in this way, they'll go for the applications. So I mean, why we are going for this present innovation, why we are taking this work means, so we have to get the advantage of both hydrogels, we have to get the advantage of nanoparticles. So why we are choosing hydrogels for these nanoparticles here? So as I said, to prevent the agglomeration, you need some polymer coating on the nanoparticle surface so that it will form the uniform throughout the surface. So yeah. So in this way, because see, I mentioned this statement importance role in the controlling the nanoparticle size. See, we are developing nanoparticle size. When you are started to accumulate all the nanoparticles singles, together, so you will get a big nanoparticle, big particle size. But in that case, there is no nano size in that. It will be like milli centimeter, something big. So we want to be keep it in nanoparticle size. The particle should be nano size. So that's why we are quoting nanoparticles. So this current research, why we are take a focus on the to control the size and shape of the particles or the matrix by entrapping our nanoparticles into the hydrogel network. So in this way, you can achieve your good nanoparticle distribution simultaneously. It will application for good application side. So yeah, so when you are going for this type of applications, in view application, like in view applications means you are directly giving it exactly to the patient. So like drug delivery, magnetic resonance imaging, and requires particle being biocompatible, stable, and biodegradable. So yeah, so so many factors we need to count into the when going for the application side. One is like particle size controlling. Another one is it should be biocompatible. See, we are directly injecting to the drug inside of our body. So even along with the drug, our nanoparticles also we are injecting into the body. So it should be non-carcinogenic. I mean, it should be biocompatible. Otherwise, these nanoparticles damage your cells so that it will trouble to your body system also. So the particles which are developed should be biocompatible. Yes, 1 minute, sir. Yeah. So yeah, further it should be stable. Stable means when you are going for the application throughout the system, that particle should be continuously it should be maintained same particles. It should not be any agglomeration, and even biodegradable also. So after certain, say, once your treatment therapy everything finish, so whatever the drug comes out of the from particles to your body, so the particle should be biodegradable. Otherwise, if it is staying in your for a long time, so that will again it will lead to some other health effects on your body. So to achieve this means to achieve the biocompatibility, to achieve the biodegradable, to achieve the stable nature. So we are treating these magnetic nanoparticles inside of the hydrogel so that it will maintain good stability, good biocompatibility nature. So how we are developing, we will explain in the next slides. Okay. Sorry. So yeah, so why? So what are the main objective of the current the main the investigation is? So novel hydrogel magnetic nanocomposites using natural polymers. So yeah, and again to develop natural that is yeah, temperature response hydrogel magnetic compounds and particle sizes analysis of the hydrogel magnetic nanocomposites, swelling of the hydrogel magnet in different media, then drug delivery studies. Yes, so many applications we are going to do. So yeah, so why biocompatible is more required? As I said earlier, so since these particles we are taking inside of the body, it should not disturb your healthy tissues. So it will be useful. It will be biocompatible so that you don't get any harmful nature from your chemical system to the body. So how we will achieve this biocompatibility? That is the biggest task, biggest challenge. See, hydrogels majority developed from the synthetic version. So if you are taking synthetic polymers and if you are you are using nanoparticles inside them, somehow it will give some problem to your healthy tissues. So that's why to reduce the toxicity of a synthetic polymer system, so we can introduce some natural polymers into your hydrogel network system so that this is a because the polymer based on the natural system will not cause any disturbance, any damage to your cells. So in this way, you can achieve your biomedical biocompatibility nature. Even if you go for the degradation studies also, this will be very useful for. So develop this. That's why we are taking this magnetic bio nano composites for different applications. So in the current scenario, in the current work, so we have taken gelatin is gelatin, which is a natural polymer. Even if you consider a lot of the capsules, whatever the tablets, that is made up of gelatin. So so by taking this advantage of gelatin, we are using this gelatin for our hydrogel system. So that it will act as a stabilizer, it will act as a biocompatible, so that you don't get any issues. So moreover, this gelatin have a lot of functional side groups. So which will go chemical cross-link. So see, as I said in the beginning, hydrogels normally itself it is a cross-linking polymer. So when you have a cross-linking polymer, and if you had if you are using acrylamide, I mean some cross-linking polymer and your gelatin also some cross-linking nature, so that it will build it will start a big cross-linking network. So the amount of holding the nanoparticles inside of the hydrogel is also very big. Even simultaneously, it will hold the nanoparticles in a proper size. So you will get both the advantages. So ultimately, the current work, I mean, what we are taking is nanoparticles, hydrogels, biocompatible system. So biocompatible system, how we are achieving by using gelatin. So which is a natural polymer. So this properties, I mean, this chemical cross-linking is a very good properties in the preparation of biometals being used as a drug delivery system or wood dressing materials also. So yeah, so how you process the experiment, how you develop this. So we have selected certain raw materials for this purpose. So acrylamide chemistry we have taken. So and a methylene bisacrylamide. This is also one type of a cross-linking system. So basically MBA acts as a cross-linking monomer. There it will start and then this TEMDA is one type of another catalyst system. Ammonia sulfate is a catalyst system. So gelatin, this iron chloride tetrahydrate, iron chloride hexahydrate, ammonium hydroxide. These are the raw materials. So if you go each individual application, acrylamide, MBA, and gelatin acts as a monomer as well as polymer system where you are converting your monomers into the polymers, and MBA acts as a some cross-linking system. So where you will get good. See, these monomers are developing. Each chain is developing. So to bridge the gap between one chain to another chain, you are using MBA. So how you are converting this acrylamide, I mean monomers into the polymers? You have some catalyst system, ammonium persulfate and TEMDA, as I mean tetramethylene diamine. So this is actually it's a type of redox reaction, I can say. So where you are converting, we are creating free radicals by reducing your for catalyst. So it will start the initiation. So how these nanoparticles are prepared? See, since we are talking about the magnetic nanoparticles, we are using iron metals as a iron metals so that we can convert into the nano iron, which acts as a magnetic because since iron is the biggest metal that will showing the magnetic nature. So ammonium hydroxide, this is used as a reducing agent to convert the iron ions to the iron nanoparticles. So so then after developing the nanoparticles, we are using some different type of characterization techniques. So whether our developer system is really polymer system, really nanoparticle system or not. So that for that we have selected some different type of characterization. Even whatever the conversion of monomer to polymer, that is also we have characterized by using FTR spectroscopy. Even the particle size, whether our particles developed inside of the hydrogels, it is in crystalline nature, amorphous. That is we can confirm with X-ray diffraction, and thermogravimetric analysis we have done for the whether this nanoparticles how much percentage of conversion you achieved in your polymer system, how much percentage of nanoparticles obtained in your hydrogel system. So for that we have taken thermogravity analysis characterization technique. Then the glass transition temperature and crystalline temperature, everything we are calculated by using differential scanning calorimetry. Then since we are talking about the surface morphology, because it should be maintained throughout the application, throughout the thing, it should maintain uniformly, it distributed. For that, so we have used both scanning as well as transmission electron microscopy for analyzing. Then whether our developed particles showing whether it is a really superconductive in nature or not, whether it is behaving in a magnetic nature or not. So that also we have studied by using VSM. VSM means vibrating sample magnetometer. Then after that, we applied our developed these nano composites for biocompatible studies and drug delivery studies. So yeah, this is the system we have adapted for developing these magnetic nanoparticles. So first what we'll take, like we explained in an materials. So we have taken acrylamide, which is available in a powder form. So we are converting acrylamide into the liquid form by dissolving in water. So in that same solution only, we are dissolving gelatin also. Gelatin is also water soluble. So we are getting one copolymer, one monomer system. Means acrylamide is monomer, gelatin is a natural polymer. So then this polymer, we are injecting TEMDA as well as persulfate, ammonium persulfate solution into the monomer copolymer solution. So then we are slowly taking into the temperature to 50 degrees C. So there, these free radicals, there you will get some free radicals. Free radicals in the sense the ammonium persulfate breaks into the, thermolytic degradation takes place, so that persulfate creates some free radicals. So these free radicals will attack into the acrylamide monomer chain. Then there will start the polymerization reaction. So that means, like I am saying, see, you are initially it is like there is no branching. So when you start the polymerization, you will get like a big molecular chain. Means your acrylamide along with the gelatin, it will start, so that all your monomer is converting into the polymer. So means it is like a redox polymerization. We use it to call redox polymerization, where your polymerization, where your monomers are converted into the polymer by redox catalyst system. So the redox catalyst system convert your monomer into the polymer system. So then we have achieved this polyacrylamide gel. So this is one type of hydrogels. It is a combination of acrylamide with combination of gelatin. So it is a copolymer system. Now we have obtained polyacrylamide. So then, what next? So this polymer is a hydrogel. So means which will hold the lot of amount of water. So what we'll do? So after obtaining the polymer, so we dissolved this polymer uh, into the iron ion solution. So what we'll do? So one side we have developed acrylamide hydrogels with combination of gelatin. Simultaneously, what we will have to do? You have to take certain concentration of both iron hexahydron, iron hexachloride solution into the dissolve into solution. You have to prepare that solution. Then you have to take this acrylamide hydrogel and you have to keep into the iron ion solution, and you have to keep for the overnight. So like you can say 24 hours. So what will happen during that time? So this hydrogel will absorb. So it will absorb the water. It will not only absorb the water, even it absorb the whatever the iron ions that are in the solution. It will absorb the iron ions also into the hydrogel. So once it is done, then this acrylamide hydrogel which absorbs the ion, it is in a swollen state. It is in a swollen state. So this we are treating with the ammonia solution. So what will happen during this? So this ammonia will convert here again some redox reaction. So what will happen? It will reduce all the ions and it will extract the electrons from the iron ions, so that you will get zero valency irons, which is called iron nanoparticles. So when you are treating ions dissolving acrylamide gelatin hydrogels, it will react, these iron ions react with ammonia, you will get hydrogen magnetic nanoparticles. So in this way, see, you are seeing this hydrogel network. So it is a copolymer system, and whatever the black particles, black dots that I indicated is a Fe3O4, means that is one type of iron nanoparticles. So what is happening here? The stabilization of Fe3 means iron nanoparticles form, simultaneously it is entrapped by your polymer network, so that it will maintain the good stabilization, good stabilization, so that it will not go for any agglomeration, so that the stability of the reaction you can get it. So this is the way how we developed acclimate gels and hydrogels. Simultaneously, you are getting this hydrogel magnetic nano composites. So yeah, so after this, see, wherever going for any application, first you have to see the how much swelling capacity of your hydrogel magnetic nano composite. So it is a simple study. So where you have to keep your once you are developing the hydrogel magnetic nanoparticles, you have to cut into the small pieces, and you have to keep for overnight, or you can keep dry for so that what all the water comes out. Then that dried hydrogels you have to keep into the uh, water, only water. You have to keep into the water. So at least for 24 hours. So since this is a hydrogel system, it will absorb the water. So why? Because why we are studying these swelling studies? Ultimately, see, if whenever you are developing your work, ultimately it should use for some selected purpose. So why you are see acrylamide already exists as a one polymer gel, but since we are taking gelatin, it whether it is giving an advantage. So since gelatin is also one type of hydrophilic polymer, so it will absorb the more amount of water. So that we are seeing uh, in this case. See, when you are varying so we have varied our formulations with not only developing hydrogels, even we have to variated our formulations by varying the gelatin percentage, MBA percentage, and everything. So we based on that, we are seeing how the hydrogels swelling studies, how the hydrogel will absorb the water. That also we are checking. So when you are seeing these graphs, so when you are varying the gelatin, so you are seeing lot of water swelling, water absorbing capacity of the hydrogel is increasing. So the same case with even MB also. But the thing is, when you are using gelatin, you are seeing some increasing trend. But when you are seeing the MBA variation, MBA also one type of cross-linking monomer. So where it will create a big bridges, so that whatever the water entrapped into, it will enhance. But here we are seeing quite opposite phenomena. So when you are taking certain molar ratiation, it is okay. But beyond that means it attained the maximum. So yeah, when but it is attain the maximum equilibrium stage, so you don't get further any swelling. I mean swelling nature of the your hydrogel. Like I said, this can we have see is there any variation in the magnetic nanoparticles? I mean, so we are this FTR spectroscopy. We are giving just for how is there any comparison between is there any difference in the spectra from regular PAM, PAM based GE, PAM based GE with combination of magnetic nanoparticles. So this is one type of fingerprint spectrum. So where you can confirm the presence of acrylamide polymer units as well as genality units and magnetic nanoparticles in your system. So if you see this green peak where it indicates the Fe3O4, which indicates the corresponding of the incorporation of nanoparticles into the hydrogel network. So yeah, so this is the X-ray diffraction pattern of the your uh, polymers as well as polymer nanocomposites. So if you see this spectrum. So it is I mean, why you are doing this both FTR and XRT diffraction, techniques. See, we are converting these iron nanoions into the iron nanoparticles. So if you have any still, if there is no conversion, if still iron ions only present into the gel network, it will be it will not useful for application. So for that, we are conferring whether is there any nanoparticles really inside of the your gel or not. So you are seeing some peak spectrum. It is indication of the nanoparticles in your gel. See, yeah. So how much if your nanoparticles content is there in your hydrogel network? So that also we are seeing means. So if you are increasing the gelatin content, it is attracting more ions, so that you are getting more nanoparticle size in your polymer network. So of course, this is if you reach a certain level, it will reach the maximum equilibrium state. Further beyond that, you don't get any nanoparticles. It is a like equilibrium state. So, see, compared to PAM hydrogels, PAM gelatin hydrogels. So when you are increasing the gelatin content, it is increasing the hydrogen content. So yeah. So how this morphology of the nanoparticles also you can see. When you see only PAM hydrogels, there is nothing. You can say very uniform surface. So when you are taking PAM and G hydrogels also, you don't see any nanoparticles inside. But when you are converting to the PAM and gelatin, I mean nitrogen, you can see the small. Yeah. So you see small areas. You will see some nanoparticles. So, so this is means the conformation of nanoparticles inside of your hydrogen network. So, addition to that, so we have studied even electron microscopy. I mean, transmission electron microscopies. So, to check the what is the exact particle size of your these things. So, we have since we are we are getting good result on the gelatin based hydrogels. We have studied G1, G4. I mean, so what is the particle size? See, our particles are coming, you can say less than 30 nanometers. This is the one type of study, magnetic purpose, I mean, VSM studies, where you can study the uh, super parametric nature. If you achieve this parabola type of structure, that indicates your particles are super parametric nature. Means it will be responsive as based responsive like a when you are going apply to some magnetic property. I mean, if you apply some magnetic field, then only it will respond. So that indicates whatever the particles, nanoparticles formed inside of the hydrogel, it is in super paramagnetic in nature. Yeah. So certain images we did, I mean, for this biocompatible studies. See, you can see biocompatible studies, how we studied some, we made some uh, uh, blood. So this is like some animal blood. So we do some compatibility studies. So if normally when you are injecting into this nanoparticles or hydro nanoparticles inside your belly, it should not affect your blood. It should not affect your cells. So if you take only PAM, PAM is it's like some type of hydrogel, but again its biocompatibility is very weak. So that's why you are seeing some blood clotting on the surface of the hydrogel. See, basically this is a hydrogel. When we dissolve it into the blood, which is along with some artificial blood, I mean blood, which is treated with some components, and then when you are dipping your hydrogel into the blood, you are seeing some clot. Means it is not biocompatible. The same case with even PAM gelatin also. But when you are dipping your hydrogel nanoparticles on the blood, see, you can't see any clotting. See, even there is no any, I mean, even the blood, whatever it is clotting, even flowing downside of the surface. So means whatever the hydrogels, nanoparticles we have prepared by combination of polyacrylamide and gel with iron nanoparticles, it is very good biocompatible, and you can use for this application different type of applications. Yeah. So this is biocompatible studies we have done. Then drug loading. So since we are talking about the targeted therapy, so we have selected one doxorubin drug, so which is one type of drug used for the cancer treatment. So so we are studied how it will affect on the with magnetic and without magnetic field. So see, if you see on the time based overall 1600 minutes we have maintained. So with magnetic field, it is releasing more amount of the drug. Means it will give lot of drug, because how it will respond when you are applying on the where you are inserting the drug inside of the hydrogel system, and when you are making this hydrogel into the water dissolving, so during this swelling nature, it will comes the drugs whatever dissolve into the hydrogel, it will comes out of the hydrogel. So in that way it releases into the body. So if you see, without magnetic field only percentage we are getting only up to 60% only, but whereas with magnetic field, so this is the more advantage of your using of hydrogel magnetic nanoparticles. I mean, so you will get more advantage of this system. So what are the basic things we have achieved? So the hydrogels we have developed for the, using free radical polymerization, characterization studies for, whatever the hydrogels, hydrogel magnetic that are developed, we also did some characterization studies. And then we have applied for the biocompatible studies and drug delivery studies. So yeah, this is the thing. So if you have any questions for next 15 to 20 minutes, I am ready to answer.
Now, vote of thanks. On behalf of our institution, I take the pleasure to extend our heartfelt thanks to Dr. N. Narayana Reddy for providing excellent information on gelatin-based magnetic hydrogel nanocomposites that triggered a promising platform for biomedical applications. We appreciate your time for contribution to cutting edge of technology. Thanks a lot, sir. Thank you for your valuable presentation.
Thank you. Thank you. It's even indeed, I'm also very happy to provide some of my research inputs during my PhD. So I'm very thankful to give an opportunity, to give a platform to explore my work.
Any questions regarding this presentation? Participants? This session will end. Maybe the second session will be start soon. Wait for some time, 5 minutes. After 5 minutes, the second session will be start the same
Yeah.
resource person.
Swami, one of my person is asking some question. What
Uh,
is the procedure of drug loading and releasing studies?
yeah,
Yeah. So basically, we did any, lab. We did some. So we prepared drug solution. So some of this drug solution in water. So we dissolved selected concentrations of the some ppm concentration of the drug, that solution. Then whatever the hydrogels prepared, that we have taken as a dry weight, and we incorporated this drug inside of the drug solution. So we kept for overnight. So during this, what will happen? Along with the water, this drug also entrapped into penetrate into the drug network. So after this, after 24 hours, we have taken this drug and we kept it for dry for the another 2 3 days, so that you will get, pure dry hydrogel nanocomposites, which is having drug inside of the hydrogel. Then for delivering releasing studies, we have prepared different types of phosphate buffer solutions. So in that phosphate buffer solutions, we have again dissolved this, dipped your hydrogel nanocomposites in the phosphate buffer solution. So each time period, we are taking selected quantity of the solution, and we are keeping for the UV studies. So where you will in UV spectra, we will giving some respective peak for the doxorubicin. So accordingly, how much drug is calculated, we are getting some quantitative analysis. So in that way, we did both drug loading and releasing studies.
thanks for Narayana sir. Explain good question. And also,
Yes,
sir,
yes,
asking me
some
some
more
other
question.
question.
Yes, yes.
Sir,
So yes,
sensor
sanitary.
pads also contain gels, but they
Yes, yes.
affect girls' health.
Yes, sir. Yes, exactly. See, polyacrylamide, actually, acrylamide is a biggest carcinogenic material. If it is in the form of polymer, there is no issue, no issue. But see, when you are converting into the monomer to polymer, definitely you have certain very ppm level of monomer also present in your polymer network. You don't get 100% conversion. So when that's why every regulatory bodies, they always maintain some certain level of monomer quantity in your polymer. So since this we call actually in chemical in scientific term it is called a super absorbent polymer. So whenever they going for they used to give for some GC studies, it should be very less ppm. Whenever something goes wrong, so this acrylamide, like I said, it is a carcinogenic. If it is in polymer, no issue. But if it's some still some ppm level of monomer will be there, that will cause some cancer issues also. That's why nowadays what we are people are doing, so they are shifting from synthetic to natural version. So like where we did incorporating some gelatin based nanopolymers in our hydrogen. So that's why we minimize the effect of toxic nature of acrylamide. So maybe because that point of view that when the people are using sanitary pads, so that will may affect the health.
Sir, I'm asking one question.
Mhm.
If the sanitary pads use the biodegradable polymer is natural polymer, but I think which is not harmful to the
But the
any
thing
girls.
is, yes. But the thing is, if you take sanitary, I mean, see, ultimately sanitary napkins main criteria, it should hold the it should hold the water content or whatever the purpose. But if you are taking only natural polymers, it don't have that much absorbance capacity. That's why still acrylamide and acrylic acid based polymer is the mode, because this acrylamide and acrylic acids will have more absorbance compared to your natural polymer. So that's
Yeah,
why it is a combination of your using.
acrylic acid and acrylamide polymers having some toxic nature.
Yes.
Good for the sanitary pads like that.
For that only. So, if you are completely, instead of taking 100% of acrylamide acrylic acid, you have to replace with, I mean, if you take 50% of this, 50% of your natural polymer you can use, then it will
Yeah.
be minimize the effect.
Yeah, these two polymers will be mixing with the blending technology. Either
Yes.
some other technology have
Sorry?
these two polymers, na, either synthetic polymer or natural polymer,
Mhm.
as a combining with the blending technology or some other technique.
It's like a blending only. So, see, natural polymer already itself, if you take wherever the polymer you are taking, natural polymer, majority 90% natural polymers are water soluble.
Yeah.
So when you are converting your polymer into monomers into the polymer, you have to insert your monomer solution into the this natural polymer solution, so that you will get a semi-interpolating network type of system.
Sorry, one more participants are asking one more question. What are
Mhm.
the analysis of blood sample taken and confirmation, sir?
The analysis means we did some UV absorption studies only. That's it. We don't go very big these things. We only UV analysis only. Means how much drug is releasing in each every time
No,
period.
no, not that much, not UV. They are
Mhm.
asking what are the analysis of blood sample taken and confirmation in the hydrogels. You showing
Blood.
one figure, na? In that figure, the blood
Uh,
absorption of
what
hydrogel.
are the, yeah, see
How to
here.
confirmation?
Yes. So we are taking here, we have taken some animal blood.
Mm,
Okay,
mm,
animal blood, but we did not take a complete animal blood. Again, what will happen if you take animal blood directly? It
mm,
will, without going for applications itself, it will be clot formation takes place.
mm,
So you take animal blood. Simultaneously, we have prepared some artificial blood also.
mm,
So we club it together so that the blood, it will expose it to outside of the environment, it will maintain for certain time. I mean, it will go for the coagulation, blood coagulation, but to prevent that coagulation, we used animal blood with a combination of artificial blood. For artificial blood, we have selected procedures in the web also, you can get it. So in that way you can prepare these things, blood samples.
mm. Thank you for your confirmation explanation. Just to share your slides and check once,
Yes.
sir.
So the second presentation is temperature responsive hydrogel magnetic composites for hyperthermia and metal absorption studies. So yeah, so what is the history behind this study? So normally, see, so far we have discussed about the hydrogels. This is also one type of hydrogels. Uh, but this is something different chemistry we have taken, different compound we have taken to develop these hydrogels. So, since we are topic is mentioning on the temperature sensor hydrogels, uh, actually what is happening? in normally in cancer therapy, when you are going for the cancer targeted treatment, sometimes you go to the little higher temperature. So uh, where your wait, yeah, so where your drug will release drastically or slowly based on your temperature system. So that's why we are selecting different type of hydrogel system for this purpose. Mainly, this is main application goes for the cancer treatment. That's why it is called lower critical solution. I mean, temperature responsive. That's why even we are given this name as a LCST. I mean, LCST means lower critical solution temperature. So this polymer, this hydrogel, behave differently below LCST, above LCST. Means certain, for example, if you take room temperature, it will below room temperature it will not behave. It will not release any drug. But if you go above room temperature, it will release some drug into the environment or whatever these things. Even the same phenomena with even water absorption studies also. Since we are talking on the hydrogels, hydrogel absorb lot of amount of water. So normally these other, if you take other hydrogels, if you take a hydrogel and dissolve in water and after keeping for drying purpose, so if you go on any normal, even lower temperature, even below that 23 to 40 or so, it will release the water. It will start to drain. But some these type of hydrogels, only if you apply give proper temperature, then only it will release the water into the medium. Otherwise, it will not release. So for this type of things, uh, instead of acrylamide, we can take an isopropyl acrylamide. So because this is having some isopropyl group at their end, so this isopropyl group will which acts as a temperature sensor, so that certain after reaching certain temperature, it will release the water. After release, if it is coming to the this, it will again shrink. So in this way, you can develop this nanopart. I mean polyacrylamide nano, poly temperature. I mean thermal responsive hydrogels. So yes, this conventional IPA is very slow response to a temperature range. So to improve the, but again, here it is also have certain drawbacks. Okay, it will release the water or drug, whatever it is holding. But the thing is, it will sometimes it will not give 100% efficiency. So to improve this response rate, we have to increase the porosity. Porosity means the whatever the cross-linking structure, cross-linking hole is there. That we have to little bit enhance the thing. Means when you are increasing the size of this hydrogel network, it will respond bit high so that you will release more amount of water. So this is the thing is so that the thermal response, thermal sensitivity of the hydrogels will be increased. So a way to achieve this is the addition of another hydrophilic monomer in the presence of NIPA, which is I mean increase. So how will achieve this porosity increasing? This you can adding you can adding you can take another hydrophilic monomer in the same NIPA network so that you can increase the LCST behavior. Further to that, since we are already talking about the hydrogel magnetic nanocomposites, so we are taking even nanoparticles also inside into this hydrogel network so that the responsive nature will be very fast. So in how we see, ultimately the advantage of NIPAM is it is a temperature responsive. But again, temperature responsive means again the performance will not be 100%. So to get it up to, I mean the responsive nature of the towards temperature system, we are again adding some hydrophilic monomer so that it will reach to certain extent. Then this again, we are developing these hydrogel nanoparticles inside of this NIPA hydrogel so that the responsive system will be very huge. So that whatever the drug incorporated inside of the hydrogel system, whatever system, it will release more amount of the drug and it will go for the exactly targeted therapy. So these magnetic hydrogels have explored hyperthermia treatment of cancer, where the consist of the raising the temperature of tumor tissue to 43 degrees C by alternating the magnetic field. So yeah, so I told you, as I said in the beginning, some of the cancer treatments will go for the even high temperature. So because uh, when you are taking the when you are incorporating the drug inside of the hydrogel and you are taking to the high temperature, so it will happen the hydrogel will like explode in means it will swell it. So whatever the drug inside of the thing, it will go inside to the body where the tissue exactly. Again, this is one type of targeted therapy. So see, why we are considering targeted therapy? Your hydrogel have some nanoparticles. So, for example, some person has a tumor in the left side of the breast. Okay, so if this, for example, to go for the breast cancer treatment. Normally, how the scenarios? So, how the cancer treatment will be? So, they use it to give drug inside of your body through some glucose bottle system. But what is happening in that case? So, this drug will affect entire of your body. You have tumor in your left side, but you are treating whole of the body. So that way what will happen, lot of the side effects of the drug will come. But when you are going for this type of system, so what they will do, they will inject the uh, drug inside of this hydrogel magnetic non-composite, which is temperature responsive. So they will exactly inject on the where the tumor is loading. So there, what will happen? They will apply some magnetic field on the. If you observe, even if you go any hospital, CT scanning or MRI scanning, what will they do? They apply some magnetic field. Like this way also in hyperthermia. So they will inject this drug exactly where your tumor loading. They will apply the magnetic field. So what will happen? You will get a dual responsive system. So one side, the nanoparticles presence inside of your body will uh, will respond. Simultaneously, by applying magnetic field, slowly the temperature also will reach to 40 degrees. So the nanoparticles as well as the gel both respond so that you will get exactly wherever the tumor is there only will get the treatment. So this is called the targeted therapy in cancer treatment. So since we are talking about the hydrophilic monomer, so for this purpose we have added one AMS monomer, which is hydrophilic in nature. So that will also absorb more amount of the nano ions into the gel so that you will get hydrogel network. So this goal of the that work is to synthesize polynipa based magnetic superhydrogels for hypertherapy and magnetic biometer for general purpose. So we can use not only for gas applications, we can use for different applications. So almost it is also similar process only what I have explained earlier. So we are taking anisopropylamide, MBA, TEMDA, ammonium persulfate, acrylamide, and these things. So yeah, see how the network forms. Uh, First, we are taking NIPAM and AMP solutions. We are blending together. Then we are treating with ammonia persulfate and TEMDA solution. See, if you are seeing this, you are getting one solid substance. So this is hydrogel. So this white hydrogel, it is a combination of NIPAM and AMPs. So once you are getting hydrogel, you're dissolving iron loads into the, so this is some orange color where iron ions loaded into the hydrogel. So then a reduction system. So what is you will once it is converted into the hydrogel magnetic nanoparticles, you will get completely black shape, black color. So means initially it was in orange color. When you are treating with ammonia, it is converting into the nano size. See, all nanoparticles normally it is in see for a magnet. Magnet, how you will see the color? It is in like a black color. So we are getting the same color here. Means the nanoparticles convert into the, I mean, the iron ions, means ions convert into the nano. So you are getting magnetic hydrogels. So yeah, again, we have repeated same swelling studies. Uh, uh, since we are taking amps in your polymer, here we are not doing any for in the first case we are not doing any swelling studies for hydrogel magnetic nanocomposites. We are did only swelling studies for the polymer. I mean hydrogels with a combination of amps. So amps polymer, wherever amps polymer is there, you are getting very very big swelling ratio. Means per each gram it is taking almost 100% of water. Means uh, so a swelling ratio of then we have selected only one type of nanoparticles. There also we have studied the uh, swelling study for magnetic nanoparticles also. Here also you are seeing some superior nature. So if you are taking hydrophilic polymer as well as nanoparticles inside of the hydrogel network, that will give the high swelling nature. This is also we are doing some uh, IR spectroscopy, which we are using for confirmation of our polymers as well as nanoparticles inside of the. So one type of characterization techniques where we did study the same similar. I mean, this is for confirmation of the nanoparticles and the formation of hydrogels. All the fingerprint regions, everything we are calculated. So here also you can see. So that 1500 sorry 1400 spectrum that will give the I mean formation of nanoparticles inside of the your hydrogel network. Yes, XRD spectra also we have confirmed. So if you see only polymer, you don't get any you don't get any Bragg's peaks. So if you take if you are taking acrylamide and Fe3O4, you are getting a lot of theta values. So which is the your nanoparticles indication. So yeah, how this nanoparticles, how much percentage formation inside of the hydrogens, that also you can confirm by the thermogrammetric analysis. Ultimately, it will if you reach to certain extent, you don't get I mean, for example, every hydrogels has a certain equilibrium position. Uh, if it is observing 100% water, it will observe only 100%. If it is observing, it is the polymer capacity is 150 grams of water, it will observe only 150. Means some threshold value is there. So here also, the formation of nanoparticles will be some threshold. Beyond that, how much iron ions you are incorporating into the hydrogel, it will not form. Am I audible?
Yeah, audible.
Yes, yes. Okay. So this I had the same. Actually, this is a scan. I mean, image for this one. Mm. Iron ion loaded hydrogel network. So since it is a salt forms, so you are seeing some salty nature on this. So this is it is, I mean, it is not converted into the nanoparticles, but when you are converting into the hydrogen nanotube, you can see some small pore sizes. So where these hydrogels are entrapped into the, sorry, where your nanoparticles entrapped into the hydrogel network. So the same. So by taking this sample, we did transmission electron microscopy images. Basically, what is the, I mean, difference between scanning electron microscopy and transmission electronscopy? Maybe some people aware or may not. So transmission electron microscopy will give the particle size of the nanoparticles formed in your hydrogels. See, in the beginning, when I started our presentation, I told you, so nanoparticles should be always uniformly distributed. Then only your objective, your application will be benefited. So if it is forming like a big clumps, you don't get much useful, much benefit. See, if you are seeing here in the transmission electron microscopy, you are seeing all the particles. What are the particles? See, uniformly distributed throughout the network. So means this hydrogel will be very good for the application. So it is, I mean, lower, see, when you are taking a lower amount of nanoparticles, you see very less amount of nanoparticles. When you are taking high amount of the nanoparticles, when your AMS acts as a good polymer, it will attract more ions, so you will get more nanoparticles inside. So means the domination for nanoparticles will be more high, so that the application side also you will get more dominance. Sorry. Okay, 1 minute. Sorry, my system was hanging. Wait 1 minute. Okay. This is also same similar studies what we have did earlier also. So this is basically why. So whether our nanoparticles are super paramedic in nature or not. Super paramedic in that sense. See, we are going for some targeted applications. Even before giving your external field, if the particles is releasing your drug, there is no use at all. So when the particles are in super paramedic nature, then only the real application, real targeted therapy will be fruitful. So I developed these hydrogels. I injected a drug inside of the hydrogel nanocomposites. I'm directly giving to the patient. So without external supply, it is directly releasing means it will be a big issue to the entire human body. So when you are applying some external field, then only these nanoparticles will respond. So the drug will release. So for that, whether these our particles are really super paramagnetic in nature, for that only we did this VSM analysis, where vibrative sampling. So where you will inject your particles, you apply some magnetic field, accordingly it will reply, so that you will get this peak. Yeah. So here in this case, we have, before not only for drug delivery studies, we have applied these hydrogel magnetic nanoparticles for some wastewater treatment purpose also. See, we have selected some wastewater. Normally, see, if you see some majority of industries, lot of wastewater effluent is coming from the industry into the municipal water or river system. It is containing lot of heavy metals and toxic metals. So if you don't purify this water, it will again comes into the our through our bore wells, and it will contaminate, so that so many health issues will come. So since this is a magnetic nanoparticle system, whatever the whatever the metals present into the industrial wastewater or normal water, you have to purify that, so that some way you can achieve the good water purity. So for that we have prepared some metal solutions. Metal solutions. Then we did the application studies. See, this is the first image is like a copper ion solution. Then we have incorporated hydrogel inside of the copper ion solution. We keep for overnight. Then we have selected this. We have collected this hydrogel. See, this polymer is a magnetic in nature. That's why it is attracting to the magnet. So you can see here more clearly. So it is attached to the magnetism. So whatever the ions presents into the copper ions, so that will attack to the gel system, so that water get purified. So that's why this is a digital camera images of the removal of toxic metal ions with hydrogel magnetic nano composites. So absorption of, then removal of magnetic nanocomponents. So then we have then the whatever the picture images that we are converting into the analytical studies. So how amount of copper we are collecting, I mean whatever the metal studies we are doing that. So we have selected for select different types of materials. So see, since you go to even room temperature also, but the thing is, since we are developing temperature based magnetic nanocomposites, so we are checking the what is the effect of I mean how this temperature affect on the hydrogel system, how the temperature affect on the hydrogel magnetic nanocomposites. See, first initially it was okay, but when reaching to 30-35 degrees C, the temperature effect is very less. But when when reaching to 40-45 degrees C, it is taking more. It started to taking again more amount of the metal ions. So means certain the LCST behavior is showing after 40 to 45 degrees C. So again, when it is going to high temperature, so if you take any polluted water and if you want to purify more your water by removing all these wasted minerals from the water, you should use this type of hydrogels. You can get more purity on this. So we have selected different types of metal systems. One is copper, one is cobalt. Yeah. So yes. Then that is for water purification studies. And this we have in the beginning we said this hyperthermia. This is called hyperthermia studies. So where your uh, drug we are not giving any drug inside of this, but sorry, 1 minute. Yeah. So we are getting some only this hydrogel dissolved in the water, and we are seeing the burst system. Burst system means breaking. Like for example, if you go take your capsule, you have you when you were inserted when you are injected. I mean, when you are taking as a medicine into your body, it will slowly swells and it will release the drug. Means whatever the drug inside of the capsule, it will comes out of the body. So that phenomena we have studied here in the form of temperature. So we are taking gels dissolved in the water, swollen gel, then uh, after that we are applying some magnetic field on the gel. So we see here the magnetic field apply means we are applying some current which will generate some magnetic field, so that gel will burst out and it will release the water amount. So here we are mentioned we have selected some concentration of the this thing. So some water, so I mean, so 10 milligrams of the water we have loaded, and we have started our experimental studies. So so in the within span of 1400 seconds, we have seen this tunnel. So compared to our NIPOM hydrogels, this Fe3O4, which is in green color, it is giving more robust nature. So means it is responding very because since it is having both nanoparticles as well as your LCSC system also, means temperature response also. See, moreover, in all the cases you can see up to 30-35 degree, it is very slow. But once it crossing 35 to 40 degree, the response is very fast. Means it is so this type of gels very suitable for hyperthermia applications. So this is first in the first graph we have compared NIPOM AMS polymer, NIPOM AMS polymers with nanoparticles. Then now we are taking to what is, I mean, see, we are, I mean, we have developed hydrogels with the different, with the variant amounts of iron particles concentrations is that. So, see, we are seeing that wherever the iron concentration, nanoparticles concentration in the gel, that is giving very robust performance. See, means if you have less nanoparticles, very less amount means in this way, when you are going for the hyperthermia, means where you are giving your drug to your body, it will, when you are applying the magnetic field, it will release the robust drug, and drug exactly applies on the tumor where it has, so that you can go for the targeted therapy applications. So yeah, so what are the basically conclusions? So hydrogel nanocomposites based on three types of natural polymers were developed. So temperature responsive hydrogel magnetic based on Hypo developed. So whatever the formation of magnetic nanoparticles in the gel network were confirmed by FTR and XRD analysis. So even the amount of the formation of magnetic nanoparticles in the gel network also identified TZ analysis. The variation in the surface morphologies of the hydrogen. I mean, this is again SEM and TEM analysis, and the VSM analysis. We can confirm whether the particles are super paramagnetic in nature. Then TEM analysis. Then we did those varieties of applications like in the previous session and this session. We did different types of application studies. So yeah, thank you. So still, if you have any questions on this topic also, I'm I'm open to.
Sir, participate in chat box. There is no asking a question, sir. Can I audible now, sir?
Yes, yes.
Sir, participate. They are not asking the question, sir. For is.
Oh, okay, okay.
It's vote of thanks, sir. Or else we have to do the any explanation, sir.
No, from my side, I'm just open for the forum if you have the questions.
Participants.
Sir, good evening, sir.
Yes.
Myself, Dr. Gurusiddhappa.
Okay.
Sir, nowadays, what is the best and cheap polymer, sir, to perform the research activities?
See, synthetic
Or,
version is always expensive. Okay.
okay,
See, basically, whatever the capsules currently our people are using, so gelatin based only. That is the cheapest polymer. Okay.
okay,
But the
okay,
thing is, when we are going for this selected type of deliveries, selected type of targeted mechanisms, see, this
okay,
is a natural polymer. See, it is
mhm,
it is like a advantage and disadvantage. Every system has a advantage, disadvantage. Okay.
okay, okay,
Some drugs are it should be give it should give in a very slowly controlled release manner. So
mhm,
if you see for gastric troubles, they are giving
yes,
coated gastric troubles. If you do direct gastric medicine to directly your stomach, it will
mhm,
damage all your stomach system. Okay.
mhm,
For that, what they are doing, they are giving, I mean, this type of synthetic polymers where it will slowly dissolve and release into your body. If you are
okay,
taking, why I'm telling, if you are considering cheap polymer, whereas gelatin is very cheap polymer. Okay.
mhm,
But the thing
mhm,
is, when you are taking immediately, it will start immediately dissolve and it will immediately start to release the drug into your body. So that will affect the your stomach system. So
okay,
it's a, I can say you can take it's a cheap polymer, but it's ultimately it should sustain your quality also. That's why it's a hybrid system is coming into the system. Means one synthetic polymer, one natural polymer, where the controlling releasing also comes into the picture, where the uh, biocompatibility is also balanced. So in that way you can achieve. But yes, there is no, I mean, you have a lot of all natural polymers, you can as cheap polymers compared to synthetic version. But again,
okay.
the results you have to be evaluated.
Sir, it is a blended between synthetic and natural polymers,
Yes,
right?
exactly.
Okay, so okay. What are the examples, sir, like blended with synthetic and natural polymers?
See now nowadays
Mm,
majority it will be combination of polyacrylamide mainly. But now
okay,
research so many I mean even polylactic acid is also coming into the because that is more biocompatibility over polyacrylamide.
okay. So percentage of this one, sir? Percentage of
You can you can take 50 to 50.
50%.
Hmm.
Okay. Sir, you used which one, sir? Synthetic or like?
No, synthetic PAM and NIPAM. For natural,
Okay.
where I am taking gelatin. Even some cases, gum macacia also we are used. Even
Hmm.
starch, cellulose.
Okay, good, sir. Thank you, sir. Have a
Yes.
nice day.
Yes.
Thank you, sir.
Thank you, sir.
Yes.
Sir,
Yeah.
for photocatalysis in wastewater treatment,
Okay.
why you choose the copper bismuth oxide, sir?
See, basically, this is just for lab studies. If you want to really studies in industry, for example, if you go for the battery, okay, when you are going for the battery industries like some Exide industry, Amran, so lot of lead is coming into the environment, but lead is more toxic. Okay. So
Okay.
you can't handle directly lead in your studies because since our labs sources are very limited, so we use only how our polymer is how we behaving towards to the metal system. That's why wherever some easy application studies is there, that's why we have selected copper, cobalt, this system. Normally, it is designed for whatever the commercially these type of systems will go for the lead. I mean, lead is very toxic. So this type we can go for the that type of studies.
Okay,
But
sir.
we are taking as a, I mean, some reference. We are taking these metals as a reference, and we did the studies. So this is some this studies has very limitations. I can say honestly.
Okay, sir. Thank you, sir.
Yes.
So, dear participant, any queries?
Added to just added to my research research. I mean, this is the we did during PhD. So just I'm adding some more inputs to these studies now. What they currently, because now I worked in both industry and academic. So now the same polyacrylamides using for everywhere in the wastewater treatment. Whatever these hydrogels
Mm.
using for wherever water treatment. For example, municipality sludge. So lot of nowadays India has a lot of municipality dust, municipality mud, everything is there. So it is a mix of mud as well as water. So we are giving our samples to the our product to the municipalities, even big municipalities, Bangalore, Hyderabad. So they have some ETP, environment treating plant. So what they will do, whatever the municipality waste come there, so our polymer they will make some solution and they will add they have very big reactors, big containers, so where they will store all this municipality wastewater in that stores. What they will do, they will add our polymer, our system into the system. What will happen, this polymer acts as a some flocculent mechanism. All mud will settle up the down and your water completely settle on the top of the surface. So what they will do, they will collect this water, they will do further treatment, biocide treatment, everything. Again, they will go for the reprocess. So like this also your polyacrylate hydrogels also helpful your for wastewater treatment.
Sir, if you don't mind, sir, have one doubt, sir.
Yes, yes.
Sir, for the wastewater treatment under the solar energy or UV radiations, UV visible radiations, sir.
Wastewater treatment. So for see for wastewater treatment, it is like a room temperature operates conditions. There is no such facilities. There is no solar treated. For example, municipalities where they will get this solar related and UV related. No, they will take this polymer. They will dissolve into the water and they will directly apply for the thing. So there is no such. I mean, you can go even with normal conditions. Also, you go for this application studies.
Okay, sir. Okay, sir. Thank you, sir.
Yes.
So now it's vote of thanks, sir.
Yes.
So myself, 𝐀𝐓𝐀𝐾, Assistant Professor in Physics from Gates Institute of Technology, Guti.
Yeah.
So on behalf of our institution, I take the pleasure to extend our heartfelt thanks to Dr. N. Narayana Reddy sir for providing excellent information on gelatin-based magnetic hydrogel nanocomposites that triggered a promising platform for biomedical application. We appreciate your time and contribution to cutting-edge technologies. Thank you a lot, sir.